So next, I'm very excited to present to you Dr. Xiaona Lu. She's coming to us from Yale University. She's an associate research scientist. She was FAST First Fellow, and she's going to be speaking to us this afternoon about the iPSC Biorepository of patient-derived neurons that has been established through FAST in collaboration with Yale that is now available to all in the community. Xiaona.
Thanks for this opportunity. I'd like to start with a question: why do we need iPSCs or the induced pluripotent stem cells in Angelman syndrome research? Animal models, such as our AS mouse model, have been incredibly valuable. For example, this little one, a 6-week-old AS large deletion mouse model. This mice has conservative regulatory elements with human. We generated this mouse with a deletion of homologous regions of human 15q11 to q13 to mimic the Angelman syndrome patients who have the 6-meg base large deletion. We can use behavior tests of this model as a readout of a therapeutic intervention to assess the efficacy, safety, and dosage response. However, despite the value, mouse models have their limitations. Human brains and mouse brains differ significantly, not just in size, but also in cellular complexity and response to the medication. This is where the iPSCs come in. iPSCs offer a way to model human brain more accurately. We're deeply grateful to have the 21 AS patients and their families who donate their blood samples to make this research possible. With their generosity, we generated 15 iPSC lines representing all the AS genotypes and normal controls, creating a powerful platform for AS research. We very appreciate our patients and their families' contribution. To develop these iPSC lines, we start by isolating blood cells, then reprogramming them into iPSCs using virus. These cells can then expanded and tested for quality. We conduct 5 key tests to ensure these cells are robust and capable of advancing AS research. We first do genotyping. We use Sanger sequencing to detect point mutation, the CGH for the UPD, large deletion, and imprinting center deficiency. Second is karyotyping. This test ensures that the chromosomes are structurally normal without unwanted rearrangement. Then we do pluripotency testing. We verify that the cells can divide into any cell types by studying cell markers and indicated this potential. And in vivo testing, we inject the iPSCs to a mouse. We can confirm that they can form teratomas with all 3 layers and further demonstrating their potential. The last one is self-renewal testing. We confirm that these cells can divide without differentiating. They also maintain their pluripotency over time. Once these iPSCs pass all these tests, they are ready for distribution. We have already shared these cells with academic institutions, pharmaceutical companies, and other partners worldwide. And we are in the process of depositing them to European Stem Cell Bank. So how do these iPSC lines accelerate AS research? iPSCs can transform into various human cell types, including neurons and even mini brains, or we call it brain organoids, that allow us to explore the disease in a human context. With these iPSCs, we can study Angelman syndrome at cellular level, test new drugs, and validate therapies initially tested in animal models. Our lab uses iPSCs in 2 main ways. First, we do 2D neuron culture, which allows us for large-scale drug screening in a fast and efficient manner. The second is 3D brain organoid culture. These organoids enable us to access the therapeutic effects more comprehensively, although it takes longer time. Here is a protocol our lab used for 2D neuron culture. Because of the high efficiency of this protocol, it only takes 10 days to get the results if we started from new iPSC cell lines, or even 7 days if the NGN2 inducible iPSC lines has been generated. I will use our ongoing RNP study to show how this works. We first use bioinformatic tools to design the SCRA. We have 17,000 SCRA were predicted. Then we score them, pick the top 20 SCRA candidates to apply to our 2D neurons. Our RT-qPCR as a result of the reactivation of UBE3A at RN level. We then perform immunostaining to assess the reactivation of UBE3A at protein level. As shown in these slides, these are the results of the first to 8 SCRAs, the 9 to 16, until the 20 SCRAs and synthesized with different Cas9 proteins. After quantification with 2 methods, we found the SCRA number 5 has the highest gene editing efficacy. Then we take this number 5 into brain organoid experiments. Brain cortical organoid culture will take at least 35 days to generate a cortical organoid from iPSCs. And it will take another 35 days to have the cortical organoid reach the mature stage. This is how our day 35 brain organoid look like. The green color represents reactivation of UBE3A 7 days after we apply the RNP at a different concentration. Red is a mature neuron marker, NeuN. Purple is a neuron progenitor cell marker, SOX2. Both these cell types have the imprinting mechanism exist. As we can see from these 6 brain organoids, along with the concentration of RNP we applied, the reactivation of UBE3A increased. If we zoom in, we could find the reactivated UBE3A expression, showing green, overlap with the 2 neuronal markers in red and purple very well. The overlap between the UBE3A and mature neuron shown in yellow, while the overlap between UBE3A and neuron progenitor cells shown in white. We also have data for mature brain organoids. We applied RNP to these brain organoids on day 63 and collect them 7 days later, that's day 70. Again, we use a green color to show the expression level of UBE3A, and also the red for mature neuron and the purple for neuron progenitor cells. We could also see the yellow ring at the outside of the brain organoids in medium and high concentrations. Then we put these organoids with organoids we cultured from control iPSC cells side by side. We could see the neuronal marker expression pattern are very similar between the bottom 2 AS brain organoid after RNB treatment and the control brain organoid. What is better, the UBE3A expression in the AS brain organoid after treatment are very close to the control organoid. In summary, we have generated 15 AS iPSC lines of all genotypes and 6 control lines with high quality. We have freely distributed these iPSC lines into investigators, academia, and industry partners. We are completing the process of depositing these iPSC lines in European Stem Cell Bank. These iPSC lines are essential and valuable resources for ongoing FDA R&D study for STEP RNP and gene editing-based AS treatment at Yale. These iPSC lines will also be very valuable to any drug and new treatment development for AS in the future. I want to close by expressing my deep gratitude to the AS patients and their families who contributed to this work. Thanks to FAST, its board members, my mentor Dr. Jiang, current past members of Jiang Lab, and also the Yale Stem Cell Center for supporting this study.
Thank you. That was great. Well, I'm sure we'll have questions on the panel.
Okay.
Thank you. So that is your work in action right there. That's what all of you went to the doctor in the middle of COVID, if you recall, to get your blood drawn so that you can send it to Yale University so they could create those organoids and those cell lines. And now they're available for anyone and everyone to use. And that is going to help grow the field to have variety, have every genotype, and ensure we're developing the best treatments for humans and not just mice living with Angelman syndrome. So thank you.